Hello everyone, I am Esteban Rodriguez at the University of Chile, and now we're talking about an extract of our course in bioorganic chemistry, focused today in the two important proteins in the body, particularly the M8 protein and oxygen transport, hemoglobin, and myoglobin. The focus on our class of today learning outcome, three important learning outcomes. The first one is the understand the paramagnetism behavior of the oxygen molecule. The second, compare the transport and storage protein to the oxygen. And finally, explain the cooperative effect observed in both proteins. The first one, we need to understand the chemical property or physical chemical properties of the oxygen molecule. The oxygen molecule is very important for the life, and they have very important features. Particularly, the electron of the oxygen molecule have the 2 ampere electron in the high energy orbital. This feature gives the paramagnetic characteristic in the oxygen. What's that mean, the paramagnetic feature? These have the oxygen could they attract to the magnetic field. For example, in the right of the slide, we can observe the liquid oxygen with the paramagnetic feature and the field they attract in the magnetic field. In comparison with the liquid nitrogen, the nitrogen don't have this paramagnetic effect, it's diamagnetic, because the all electron is parade in the orbital, in don't feel the affinity to the magnetic field. So oxygen therefore oxygen requires a specialized transition method when considering this characteristic to capable to single electron transfer to bind and transport it if effectively. And now appear the most important protein on the body, myoglobin and myoglobin, both protein, the principal metal center to transport and storage oxygen. What feature have this protein structure? Particular, both proteins have the N group. This N group, considering the metal center, this metal center is iron. Iron in oxidation state number 2. This sun riding in the plane with the porphyrin rings, the stabilizes in the equatorial side the metal ion in this step. In the bottom or in the down, depending on you observe the protein, we have the metal ion is unit coordinate is the perfect concept. Unit the histidine ratios they connect the metal center with the complete protein structure. In the other position in the up in this image is completely free. In this position, the oxygen can unite to the metal center. So now we can observe the principal structure difference in between the both proteins. In the left, we can observe the myoglobin, the principal function in the intracellular storage in the muscular tissues and capture oxygen locally. This protein have we can see the circle have the only one M group. M group is the metal center, also running the porphyrin rings, and this protein have one M group around 153 amino acid residues. In the right, we can observe the hemoglobin. This protein has the principal function systematic blood transport and represent around the 65% in the bodily iron. This structure you can observe for cycles because this protein have the 4N group. This presence of the 4N group or 4 metal ion center have the ability to transport the oxygen in the blood. Why this protein can storage or transport the oxygen? Because we can observe the different act in the transport oxygen process. When the protein is deoxygenated without oxygen, the metal center have in the high spin with oxidation state number 2. So writing the porphyrin rings into the equatorial side, in the up, the histidine ratios connect to the rest of the protein. In the below is completely free. In this zone can unit the molecular oxygen molecule. If we can observe the geometry around the metal center, we can observe a square-based pyramid with the below position completely free. When the protein unit the first oxygen molecule, this molecule occupies the free position in the below in this scheme. When the oxygen molecular unit the metal center, the first change, the metal center change the spin from the high spin to the low spin, and then change the oxidation state from 2 to 3. And the oxygen molecule, if you remember, have the 2 unpaired electron, give 1 electron to the metal center, generating a superoxide unit to the metal center. This process, in particular in the myoglobin, generate a power cooperative effect. What does it mean? When the unit, the myoglobin, the first molecular oxygen molecule, generate change in the geometry around the metal center, and then this chain of the protein structure generate conformational chain until the second metal center of the second end group. And this center have the very high ability to unite a new oxygen molecule. This effect generate a new conformational chain in the protein and generate the third metal ion, very high ability to unite the third oxygen molecule. And finally, the fourth metal center have the very highest ability to unite the oxygen molecule, and this reason can the protein hemoglobin have the ability to unite until 4 oxygen molecule in very fast transport to the metal to the body. So in the next class, we can understand this graph in which discuss in the class in the power working what is the difference or what is the feature in the oxidation saturation percent pressure of the oxygen in the body, and how we can understand the myoglobin or hemoglobin can act to transport or to storage the oxygen. Thank you in advance for this class and see you in the next class. Thank you.